Welcome to our latest video, which has the title Revising Amino Acids. This video is suitable for A-level students. By the end of this video lesson, you should be able to describe the general formula and classification of alpha-amino acids. You should also be able to explain the amphoteric and Zvita ion nature of amino acids and the effect on melting temperature and solubility. And finally, you should be able to understand how alpha-amino acids can be combined to form a dipeptide. So let's start this video by looking at the structure of an amino acid. Now, amino acids are carboxylic acids that have an amino group, an NH2 group, as part of the molecule. And the term alpha amino acid is used to describe an amino acid which has the NH2 group, the amino group, bonded to the same carbon atom that contains the carboxylic acid group. Now, this carbon is called the alpha carbon, as seen in this diagram. So here with this molecule we have an NH2 group, the amino group, bonded to a carbon, which we call the alpha carbon, and on that carbon will be a hydrogen and a group that varies, we're going to call it R here, and a C double bond O, OH. Now, the two simplest amino acids are 2-amino ethanoic acid, which is commonly known as glycine, and 2-amino propanoic acid, which is commonly known as alanine. And you can see I've drawn the structures of these two amino acids on this slide. Now, when we name an amino acid, the carbon in the carboxylic acid group is carbon number 1. So here we have glycine 2-amino ethanoic acid. The carbon in the carboxylic acid group is carbon number 1. So therefore the amino group is on carbon 2. That's why it's 2-amino ethanoic acid. And if we look at 2-amino propanoic acid, if the carbon in the carboxylic acid group is carbon number 1, we have two other carbons present, that's why it's propanoic acid, and the amino group, the NH2 group, is bonded to the second carbon in the chain. Now, previously at A-level, you would have covered optical isomerism. And if you want to revise optical isomerism, please check out our earlier video on this. Now, optical isomerism is a form of stereoisomerism, where you have the same molecular formula, the same structural formula, but a different arrangement of atoms in space. And optical isomerism occurs when you have a carbon next to 4 different groups. And when this happens, we call it a chiral center. So all the amino acids except for glycine have a chiral center because they've got a carbon next to 4 different groups. Now, optical isomers have virtually the same properties as each other, apart from their ability to rotate plane polarized light. Now, optical isomers are called enantiomers, and they are non-identical mirror images of each other, which mean that they are mirror images that cannot be superimposed one on top of the other. So to test your understanding of optical isomerism, we want you to attempt this practice question. So read through the question, pause the video, have a go at it, and then we'll go through the answers. So in this question, you're asked to draw the pairs of optical isomers, marking in their chiral center with an asterisk for alanine and valine. So you can see I've marked in the chiral center here, and I'm going to draw a tetrahedral. So I'm drawing 2 bonds here in the plane, a triangle to represent a bond coming out of the plane, and a dashed line going behind. And I'm now going to circle the 4 groups attached to the chiral center, and I'm going to transfer these groups onto my tetrahedral. So I have a CH3 here, I have a C double bond O, OH, and I have an NH2 and a H. So I'm putting those groups onto my tetrahedral. I've got a dashed line to represent a mirror, and all I then need to do is draw another tetrahedral, and I'm drawing it so it's a mirror image of my first tetrahedral. So I'm going to put in the 2 lines in the plane, the triangle representing a bond coming out, and a dashed line representing it going behind. And then all I need to do is put the same groups So then I just put my CH3, my COOH, my NH2 group and my H onto the tetrahedral so that they line up opposite the groups on my first tetrahedral. So then I have non-identical mirror images of each other, and these are enantiomers, and they would rotate plane polarized light in opposite directions. So now I'm going to do exactly the same for valine. So first of all, I have to identify the chiral center, the carbon next to 4 different groups, and that's the reason that there is optical isomerism here, because there's a carbon next to 4 different groups. So I've marked that in with an asterisk, and then I'm going to draw a tetrahedral, and I'm going to put the 4 groups attached to the chiral center. So here we have a C double bond OH, a H, an NH2, and a CH3, CH, CH3. And these are going to be put onto the tetrahedral. So I'm going to put in the CH, CH3, CH3. I'm going to put it there, and then I'm going to put the other groups around the tetrahedral. So I'm going to put a C double bond OH, and then an NH2 and a hydrogen. So all I then need to do is draw my other tetrahedral, and I'm drawing this so it's a mirror image of my first tetrahedral. So I'm going to draw in my tetrahedral now and put in the groups. So I'm going to put a CH, CH3, CH3 group in. So I'm putting that in now. And then I'm just going to put a COOH group up top here. And then the bonds coming out and going behind are going to have an NH2 and a H, and I'm going to line them up opposite the NH2 and H groups from the first tetrahedral. So I now have 2 tetrahedrals to represent the 2 optical isomers that would rotate plane polarized light in opposite directions. Now, amino acids have some quite unusual properties. For example, they are crystalline solids at room temperature, and their melting points are much higher than you might expect for molecules of their size. And this is because they exist in what we call a Zvita ion form. Now, amino acids contain a carboxylic acid group and an amino group, and the carboxylic acid group is acidic and the amino group is basic. So the carboxylic acid group can donate a H plus ion to the amino group. The carboxylic acid is therefore acting as an acid, and the amino group, the NH2 group, acts as a base because it's a H plus acceptor. And when this takes place, we have a Zvita ion that's formed. So I've drawn here an amino acid with an NH2 group, a basic group, and an acidic group, a COOH group, and you can see that the acidic group, the COOH, gives H plus to the NH2 group, and we form what we call a Zvita ion, where we have an NH3 plus group on one side and a COO minus group on the other. Now, the formation of a Zvita ion, where you have a positive NH3 group and a negative COO minus group, means that you have strong ionic forces that can exist between Zvita ions. You can have strong electrostatic attraction between a negative group on a Zvita ion and a positive group on another Zvita ion. So there are strong ionic bonds, strong ionic forces that exist between Zvita ions, which takes a lot of energy to break, and that results in high melting points. And you can have amino acids that have melting points around 200 to 300 degrees C, which is a lot higher than you would get for organic molecules of the same size. So that is a really unusual property, and that's caused by the ionic forces, the ionic bonding that exists between Zvita ions. Now, in addition to the higher melting points, Zvita ions also affect solubility. Now, because you've got the presence of these positive and negative ions in a Zvita ion, it means that amino acids are generally soluble in water and insoluble in non-polar organic solvents, such as hydrocarbons. And this should be no surprise, really, because you've seen previously that ionic compounds can dissolve well in water because water can form hydrogen bonds to the positive and negative ions. So solubility of amino acids tends to be quite good in polar solvents, except if the amino acid contains large non-polar groups, such as long hydrocarbon chains or a benzene ring, which would therefore reduce the solubility. So now let's explore the amphoteric properties of amino acids. Now, amino acids are classed as amphoteric as they react with both acids and bases. So I'm going to draw now an amino acid. I'm going to draw glycine. So I've put in my amino group and my carboxylic acid group, and I'm going to convert this to a Zvita ion. So a Zvita ion is formed when the carboxylic acid group loses a H plus ion, and this H plus ion is accepted by a lone pair of electrons on the nitrogen of an NH2 group. It's a coordinate bond, and we end up with NH3 plus, C double bond O, O minus, and we have a Zvita ion form. Now, a Zvita ion only exists at a certain pH, and that pH is called the isoelectric point. And if we go either lower pH than the isoelectric point, or we go to a higher pH than the isoelectric point, the Zvita ion no longer exists. So I'm going to show you what happens if we go to a lower pH than the isoelectric point. So let's say the isoelectric point is 6.2 pH, and we go lower than that, so we went to say 5, we would be adding H plus ions. And the Zvita ion would convert to what I've drawn here, because the H plus would add to the C double bond O, O minus to form C double bond O, OH, and we would be left with the following ion that just has a positive charge. So the Zvita ion only exists at a certain pH. Now, if we go to a higher pH than this isoelectric point, the pH that the Zvita ion exists, we do this by adding OH minus ions. So if we add OH minus ions to my Zvita ion, the NH3 plus group would react with this OH minus. A hydrogen would be ripped off that group, and we would be left with an NH2 group and a C double bond O, O minus ion. So my ion would just have a minus charge. So you can see that amino acids are classed as amphoteric because they react with both acids and bases. And that's what the term amphoteric means. It means they react with both acids and bases. They have acidic and basic properties. So the last thing we're going to look at in this video is the formation of dipeptides. Now, amino acids can join together to form peptides via an amide or peptide link. And a dipeptide is formed when two amino acids join together, because di means two. And when two amino acids join together to form a dipeptide, a water molecule is eliminated. So we're going to look at now how alanine can join via a peptide linkage to the amino acid valine. So I've got alanine here and valine. And what I'm going to do is I'm going to join them together via this peptide linkage. So the way it works is that the OH from the carboxylic acid group here is going to join with the H from the NH2 group of the second amino acid, and we're going to form water, and that's going to be eliminated. And then I'm just going to draw the resulting dipeptide. So we're going to have NH2 bonded to a carbon, bonded to a H, bonded to a CH3, bonded to a C double bond O. Now the OH has been eliminated, so the C double bond O is going to be bonded now to a nitrogen with just one hydrogen on it, and that's a NH from valine, and I'm going to put in my other groups. So I've got a CH, a CH3, a CH3 here, and then this is bonded to a carbon next to a hydrogen and a C double bond O, OH. So that would be my dipeptide, and that is a dipeptide formed when alanine is my first amino acid and it's joined to valine. Now I'm highlighting now the peptide or amide link. That's the C double bond O NH group, and the bond joining them together is called a peptide bond. So that bond between the C double bond O and the NH is a peptide bond, and I've drawn alanine next to valine. Now I could have drawn valine as the first amino acid joined to alanine, and that would be a second possible dipeptide that I could have here. Now it's important to mention that this video is only covering the formation of dipeptides, and if you joined 3 amino acids together you'd form a tripeptide, and if you join lots and lots together you'd form a polypeptide, which is commonly known as a protein. Now a protein will have somewhere in the range of between 50 and 2,000 amino acids joining together in its formation, and the formation of proteins and tripeptides won't be covered in this video. So now let's test the concepts covered in this video with some practice questions. So this practice question is in a number of parts, and here's the first part. So please read through the question, pause the video, have a go at it, and then we'll show you the second and third parts of this question. So here's parts 2 and 3 of this question. So read through the questions, pause the video, have a go at it, and then we'll go for the answers. So in the first part of this question you have alanine, 2-aminopropanoic acid, and it says it has a melting temperature of 258 degrees C, whereas a similar compound, 2-methylpropanoic acid, melts at -46 degrees C, and they've drawn both compounds here, and they're asking you why the value for alanine is so much higher than 2-methylpropanoic acid, and it's a 2-mark question, but there's actually 3 points that we would recommend you make here. So alanine forms a Zvita ion. Forces between alanine molecules are ionic bonding, and ionic bonding is much stronger than the intermolecular forces you would have in 2-methylpropanoic acid, which are hydrogen bonds and van der Waals. So there's 3 really important points here, but in this exam question they only gave you a maximum of 2, and you could have any 2 of those points for the 2 marks. So for parts 2 and 3 you have to draw the 2 possible dipeptides that can form when 1 molecule of glycine combines with 1 molecule of alanine, and then you have to circle the peptide linkage in one of your dipeptides. So alanine can be bonded to glycine, and alanine can be the first amino acid and glycine can be the second when they join together in a dipeptide, and I could also have glycine as my first amino acid and then alanine. So here I'm going to draw alanine as the first amino acid bonded to glycine, because that's what it is in the question, and I'm just going to remove my water, and I've circled that, and I'm just going to join them together after water's removed. So I have an NH2 bonded to a carbon which has a CH3 and a H, and I have C double bond O. I've lost the OH, so it's going to be bonded to an NH, and then I'm going to have a CH2 and a C double bond O OH. So water's been eliminated here, and that would be my first dipeptide, and that would be alanine bonded to glycine. So my second dipeptide would be a glycine molecule as my first amino acid joining to alanine. So we have an NH2 group here attached to a carbon which has 2 hydrogens on it, and we have a C double bond O OH. So I'm drawing glycine first, and then I'm going to draw in my alanine molecule, which is a carbon bonded to a CH3 here and a hydrogen, and on this carbon we have an NH2 group, and we have a C double bond O, OH. So all that happens then is a OH group is removed from the carboxylic acid group of glycine, a hydrogen is removed from the NH2 group of alanine, we form water, and a peptide or amide linkage forms in between the two amino acids that joins them together. So we have NH2 bonded to a carbon, which is attached to two hydrogens. That is bonded to a C double bond O, which is bonded to an NH. And then we have a carbon, which is attached to a CH3, a hydrogen, and a C double bond O, OH. And that is my second dipeptide, where we have glycine attached to alanine, and glycine is my first amino acid and alanine is my second. And then all I need to do is circle the peptide linkage in one of the dipeptides. So I'm going to circle this now. It's a C double bond O, NH, and the actual peptide bond is this bond between the C double bond O and the NH, and that is the peptide bond, and I've circled the peptide linkage. So there's 2 marks here for the dipeptides and 1 for circling the peptide linkage. So that concludes this video lesson. So after watching this video, you should now be able to describe the general formula and classification of alpha amino acids. You should be able to explain the amphoteric and Zvita ion nature of amino acids and the effect on melting temperature and solubility. And finally, you should be able to understand how alpha amino acids can be combined to form a dipeptide. So that concludes our video. Please check out our YouTube channel, Dr. O Chemistry, and our Twitter site, which contains lots of chemistry information and links, @RadaChemistry.